So hello, my name is Salty. Welcome to Steady Bro, and I'm your bro for life. Today we'll be looking at the regulation of glycolysis, as I said in my last video. Glycolysis is a very important metabolic pathway, and as such is tightly regulated. During my last video, I made mention of a couple of enzymes which I said were quite important in the regulation of glycolysis. As a rule of thumb, in metabolic pathways, the enzymes that catalyze irreversible reactions are normally the ones that regulate said metabolic pathway. In the case of glycolysis, hexokinase, fractokinase, and pyruvate kinase. Let's do a quick recap. Hexokinase was that enzyme that catalyzed the conversion of glucose to glucose 6-phosphate and hence trapping it within the cell. Phosphofructokinase was that very enzyme that catalyzed the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. You remember that? Then we have pyruvate kinase. Pyruvate kinase converted phosphoenol pyruvate to pyruvate. These three enzymes regulate glycolysis. In this video, I'll be looking at how glycolysis is regulated in the muscle. The subsequent one, we shall look at how it is done in the liver. The muscle is quite a gelatinous tissue. It consumes a lot of energy, mainly because of its function. All these contractions it does. When we do look at the physiology of muscle contraction in a future video, this will be made rather clear. Because of the muscle's dependence on ATP, its glycolytic requirement is based on the ratio between ATP and AMP, the balance thereof. How does this impact phosphofructokinase, hexokinase, or pyruvate kinase? We shall begin from phosphofructokinase. When there is an abundance of ATP within the muscle tissue, ATP allosterically inhibits phosphofructokinase. Allosteric inhibition is when an enzyme is inhibited not on its active catalytic site where its natural substrate binds, so not where fructose 6-phosphate natural substrate of phosphofructokinase. So normally fructose 6-phosphate binds this catalytic site of phosphofructokinase and is converted to fructose 1,6-bisphosphate. The allosteric inhibition by ATP is not on the active site or catalytic site, is done somewhere else. When this allosteric inhibition occurs, it causes a conformational change in phosphofructokinase. When we say conformational change, it changes how the different parts of the phosphofructokinase enzyme interact with each other. If normally it is like this, if an ATP binds it allosterically, it becomes something like this. So it no longer has the same affinity it had for fructose 6-phosphate, and hence it becomes less active. Glycolysis cannot flow. It does not take place. It is inhibited. It is blocked. Barrier. It is blocked. I hope this is making sense. However, when the balance between ATP and AMP is low, i.e. when ATP is low and AMP is high, then this allosteric inhibition is reversed and the enzyme phosphofructokinase regains its affinity for fructose 6-phosphate and hence it can convert it to fructose 1,6-bisphosphate and glycolysis can flow. I hope this is making sense. AMP is an adenosine with one phosphate. Within the muscle, when the pH falls, this potentiates the inhibitory activity of ATP on phosphofructokinase and hence stop the flow of glycolysis. A balance between ATP and AMP and not ADP is used to promote glycolysis or to stop glycolysis for two reasons. When ATP is being utilized within the cells, adenylate kinase can salvage, takes one ADP, another ADP, brings them together, forms ATP and AMP. Having a certain amount of ADP does not really tell you the energetic status of the tissue. To be more sensitive to the changes in ATP requirement, AMP is better suited. The second reason is that naturally there's more ATP than ADP and there's more ADP than AMP within the tissue. So if I make a small change in ATP, the consequent changes that will trickle down will be magnified in terms of the AMP pool within the cell. Hence, using AMP as a signal, not ADP, makes fine-tuning of glycolysis and its regulation more accurate, more sensitive. Let's look at how hexokinase plays a role in the regulation. When phosphofructokinase is allosterically inhibited by ATP or a fall in pH, fructose 6-phosphate levels rise, because it is not being converted to fructose 1,6-bisphosphate. When fructose 6-phosphate levels rise, so does glucose 6-phosphate, because there is an equilibrium, and hence glucose 6-phosphate is not being converted into fructose 6-phosphate. Now, a rise in glucose 6-phosphate levels acts as a negative feedback loop to hexokinase. It inhibits the activity of hexokinase because it's telling the cell there is no need conversion of glucose to glucose 6-phosphate, and that is controlling glycolysis because we do not have any more sugar coming into the cell and the sugar remains in the blood. From this, it's quite obvious that phosphofructokinase runs things around you and not hexokinase. This is mainly due to the product of their respective reactions. Hexokinase converts glucose into glucose 6-phosphate. Fructose 1,6-bisphosphate is the product of phosphofructokinase. Glucose 6-phosphate is not a unique intermediate to glycolysis. Fructose 1,6-bisphosphate, on the other hand, is a unique intermediate to glycolysis. Glucose 6-phosphate can be used to produce glycogen, and hence it makes sense for phosphofructokinase and its reaction, the fructose 6-phosphate to fructose 1,6-bisphosphate, to be the committed step, the rate limiting step upon which the regulation of glycolysis is based, not hexokinase. Let's look at pyruvate kinase. Pyruvate kinase is the enzyme that catalyzes the reaction from phosphoenol pyruvate into pyruvate and ATP. Because the end product is ATP, when the energy charge within the muscle tissue is high, i.e. when there is an abundance of ATP within the muscle tissue, ATP allosterically inhibits pyruvate kinase like it did phosphofructokinase. Once again, an allosteric inhibition means ATP binds pyruvate kinase at a different site from its active catalytic site, causing a conformational change which decreases its affinity for phosphoenol pyruvate and hence becomes less active and decreases the flow of glycolysis. I hope this is making sense. When the charge, however, is low and there is the need for glycolysis to go on to produce energy, fructose 1,6-bisphosphate promotes the activity of pyruvate kinase and more ATP is produced. This concludes the regulation of glycolysis within the muscle. In the next video, we shall be looking at how glycolysis is regulated within the liver and how this differs from that which occurs in the muscle. I hope this was useful. If there is anything you'd like me to improve upon, just make me aware of it. I'm Steady Bro, and salute!